All right, ready to tackle a bug that keeps even the most experienced medical microbiologists on their toes.
You're talking about TB?
Yeah, tuberculosis. Talking about an ancient adversary, right?
Absolutely. It's been around for, well, ages.
No,
and treating it's not a walk in the park.
Definitely not. And that's why we're diving deep today into Chapter 13 from Walsh and Wentwich's Antibiotics. Challenges, mechanisms, opportunities.
Yeah, good old Walsh and Wentwich.
Right. The chapter's called Tuberculosis, a formidable challenge for antibiotic therapy, which honestly might even be an understatement.
You're telling me?
So today we're zeroing in on TB antibiotic therapy. Yeah, the stuff that really matters on the front lines for folks like you.
Makes sense. And speaking of front lines, TB has been waging war on humanity for a long, long time.
Oh, for sure. But how long exactly?
Well, it likely originated from domesticated animals way back like 10,000 years ago. And get this, a whopping 2 billion people are walking around with latent TB infections.
2 billion. Wow, that's a huge chunk of the world's population.
It is, almost a third, actually.
Really puts the global impact of TB into perspective. But what makes it so darn hard to treat? The chapter points to M. tuberculosis's ridiculously tough cell envelope.
Ah, yes, the mycobacterial fortress.
Exactly. It's like those medieval castles with walls you just can't breach.
Well, picture this. Oh, it is. And it gets even sneakier. It forms these clusters called granulomas where it can basically lie dormant for years, sometimes decades.
Decades. Seriously.
Seriously. So it's not only tough to get to, but it can also just disappear, making treatment a nightmare.
Wow, talk about playing hide and seek with our immune system. So how do we even begin to fight something so elusive? Brute force.
Not quite. Brute force alone won't cut it. It's more like a strategic chess game. We need precision and coordination, which is why the standard treatment involves a combination of 4 first-line drugs.
4. Okay, let's hear them.
You've got isoniazid, rifampin, pyrazinamide, and ethambutol.
Got it. So it's a multi-drug cocktail? Why not just one super powerful drug?
Well, the combination therapy is crucial for minimizing resistance. Hitting the bacteria with multiple drugs at once makes it way harder for them to develop resistance to all of them simultaneously.
Oh, I see. So it's like attacking from multiple fronts, making it harder for the bacteria to adapt and survive.
Exactly. Now, each of these drugs has its own unique mechanism of action, which is what makes the combination so effective. Let's start with rifampin. It directly binds to the bacteria's RNA polymerase.
RNA polymerase, that's the enzyme responsible for transcribing DNA into RNA, right?
You got it. So rifampin basically shuts down protein synthesis, which is essential for the bacteria to function and survive.
Okay, that makes sense. No proteins, no life. What about the other 3? The chapter mentioned something about them being prodrugs.
Yes, you're right. Isoniazid, pyrazinamide, and ethambutol are prodrugs, meaning they need to be activated inside the bacteria to work their magic.
Ah, like sleeper agents infiltrating the enemy camp.
I like that analogy. So once activated, isoniazid goes after the synthesis of mycolic acids. Remember those long-chain fatty acids that make the cell wall so tough?
Yeah, the fortress bricks.
Exactly. Isoniazid inhibits the enzyme responsible for making those mycolic acids, essentially weakening the cell wall.
So it's like taking a sledgehammer to that fortress wall.
Yeah.
But how does isoniazid actually work?
It gets a bit technical, but basically it forms an adduct with NAD, a coenzyme that's essential for many metabolic processes.
NAD, nicotinamide adenine dinucleotide.
You got it. By binding to NAD, isoniazid basically sabotages the enzyme responsible for making mycolic acids. And there's a fascinating parallel with another drug, pyridomycin. It targets the same enzyme, but acts as what we call a bisubstrate inhibitor, meaning it mimics both the enzyme substrate and NAD, making it even more effective.
So it's like a double agent fooling the enemy twice.
You could say that. And then there's ethionamide, which works in a similar way to isoniazid, but is activated by a different enzyme, adding another layer of complexity to this whole biochemical battle.
Wow, this is getting pretty intricate.
It is. And to round out our first-line arsenal, we have pyrazinamide and ethambutol. Pyrazinamide is a bit of a mystery. We know it messes with protein synthesis and energy production, but the exact mechanisms are still being figured out.
Okay, so we know it works, but not exactly how. And what about ethambutol?
Ethambutol inhibits the formation of arabinogalactan.
Ah, right. Back to that mortar that holds the fortress wall together.
Exactly. So we have 2 drugs attacking the cell wall's building blocks and one going after the mortar.
That's quite a coordinated assault.
Yeah.
But I imagine even with all these weapons, TB can still put up a fight, right?
Oh, absolutely. Sometimes these first-line drugs aren't enough, either because of resistance or other complications.
Okay, so what happens then? We call in the reinforcements.
Exactly. That's where second-line drugs come in, offering more options, but often with more side effects and challenges.
All right, well, before we dive into those second-line therapies, let's see if you've been paying attention. Ready for a pop quiz?
Bring it on. I love a good challenge.
Great. First question, why is combination therapy so crucial for treating TB? Think back to what we discussed about bacterial evolution and survival strategies.
Well, it all comes down to minimizing drug resistance, right? Using a multi-drug cocktail with different mechanisms of action makes it a whole lot harder for M. tuberculosis to develop resistance to all of them at the same time.
Right, like setting up multiple traps. Makes it nearly impossible to escape. Okay, question number 2. Which first-line drug directly inhibits RNA polymerase, effectively shutting down protein synthesis?
That would be rifampin. It's the one that doesn't need any activation inside the bacteria. Goes straight for the transcription machinery.
Bingo. Okay, shifting gears a bit. Question 3. What's that unique characteristic of the mycobacterial cell envelope that makes it such a formidable barrier for antibiotics?
It's that waxy multi-layered structure we talked about. The combination of those long-chain mycolic acids and the arabinogalactan scaffold that links them together just makes it incredibly tough for drugs to penetrate.
Right, like a microscopic fortress.
Yeah.
Okay, time for a bit of a thinker. Question 4. Isoniazid is often described as a prodrug. What does that mean? And how does its activation contribute to its effectiveness against M. tuberculosis?
So a prodrug is basically inactive until it's metabolized within the target cell, right? In the case of isoniazid, it needs to be activated by a bacterial enzyme called KG. And this activation process leads to the formation of an adduct with NAD, which is, well, essential for a bunch of metabolic reactions.
Yeah, NAD, we talked about that earlier, nicotinamide adenine dinucleotide.
Right. So by binding to NAD, isoniazid basically blocks the synthesis of mycolic acids, those essential components of the cell wall.
So it's like a Trojan horse sneaking in and then disrupting things from within.
Huh.
Okay, last but not least, question 5. Besides directly inhibiting mycolic acid synthesis, what other mechanisms are being explored to disrupt the integrity of the mycobacterial cell wall?
Well, ethambutol already targets arabinogalactan synthesis, right? But researchers are always looking for new ways to interfere with cell wall assembly. Some are investigating inhibitors of the enzymes involved in transporting and assembling those mycolic acids, and others are looking at ways to disrupt the formation of peptidoglycan, another key component of the cell wall.
Wow, so it's a multi-pronged attack on the fortress, targeting not only the building blocks, but also the construction process itself. Fascinating stuff. I bet we'll be hearing a lot more about these novel approaches in the near future.
Oh, for sure. The search for new ways to dismantle the mycobacterial defenses is definitely ongoing. But now, let's circle back to those situations where the first-line defenses aren't enough. Time to call in the reinforcements.
Right, the second-line drugs. So, tell us, what are the key players here and how do they differ from those first-line options?
Well, the second-line drugs are a pretty diverse bunch. They have different mechanisms of action, but they tend to be, well, less effective or have greater side effects. That's why they're usually reserved for those tougher cases.
Makes sense. Like bringing in the special forces when the regular troops encounter heavy resistance. So give us some examples of these second-line heavy hitters.
One group you've probably heard of is the aminoglycosides, things like streptomycin and amikacin.
Yeah, those ring a bell.
They work by binding to the bacterial ribosomes and messing up protein synthesis. But their effectiveness depends on the bacteria's energy state.
Energy state, what do you mean by that?
Well, aminoglycosides need energy to be transported into the bacteria. If the bacteria are in a low energy state, well, they just don't take up as much of the drug, and that can make the treatment less effective.
Ah, I see. So these sneaky bacteria can basically power down and become less vulnerable to the drugs.
Precisely. And that's a big issue with M. tuberculosis because it can persist in this low energy state, especially within those granulomas. Makes it harder to target.
Clever bacteria, but frustrating for us. What other options do we have in this second line of defense?
Another interesting one is para-aminosalicylic acid, or PAS for short. It's an older drug, but it still has its uses. PAS is a structural analog of para-aminobenzoic acid, Kba for short, and PABA is essential for folate synthesis.
Folate synthesis. Now, remind me why that's important again.
Folate is a B vitamin that's crucial for DNA synthesis and all sorts of metabolic processes. By mimicking PABA, PAS basically disrupts folate synthesis in the bacteria, slowing down their growth.
So it's like starving them of essential nutrients. A different approach, but effective nonetheless. What else?
Then we have the fluoroquinolones, drugs like levofloxacin and moxifloxacin.
Ah, yes, the DNA gyrase inhibitors.
Right. They inhibit both DNA gyrase and topoisomerase IV, which are essential for DNA replication and repair. So by targeting these enzymes, fluoroquinolones block bacterial DNA synthesis, leading to cell death.
Got it. Another way to sabotage their replication machinery. What else do we have in our arsenal?
We also have cyclic peptides like capreomycin. They also target the ribosomes, but at a different site than the aminoglycosides. They interfere with protein synthesis, but their exact mechanism isn't completely clear yet.
Okay, so a pretty diverse set of backup options, each with its own way of attacking the bacteria. But just like with the first-line drugs, resistance is a big concern, right? How does M. tuberculosis develop resistance to these second-line drugs?
That's where things get really interesting. Unlike a lot of bacteria that acquire resistance through horizontal gene transfer, kind of like trading resistance genes like baseball cards.
I love that analogy.
Tuberculosis mostly develops resistance through spontaneous mutations in its own genes.
So it's more about internal evolution than external acquisition.
Exactly. And because these mutations are random, they can pop up in the genes that encode the drug targets, or in genes involved in drug activation, or even in efflux pumps.
Efflux pumps?
They're like little pumps that bacteria use to pump out toxins, including antibiotics. So a mutation in a gene related to an efflux pump can make the bacteria more efficient at getting rid of the drug.
Sneaky. So basically, the bacteria are constantly evolving ways to outsmart our drugs. It's an arms race.
That's a great way to put it. And that's why we're seeing these scary acronyms like MDR-TB and XDR-TB.
Right, those don't sound good. What do they stand for again? And why are they so concerning?
MDR stands for multi-drug resistant TB, meaning the bacteria are resistant to at least isoniazid and rifampin, our two main first-line drugs. And then there's XDR-TB, which is even scarier. It stands for extensively drug-resistant TB. That means resistance to even more drugs, including the fluoroquinolones and at least one injectable second-line drug.
So basically, fewer treatment options, longer treatment times, and a higher risk of treatment failure. Not good at all. But there's got to be some hope on the horizon, right? What are some of the newer approaches being explored to tackle these resistant strains?
There's definitely hope. Researchers are working hard to develop new drugs and strategies. One promising avenue involves a combination of meropenem and clavulanate.
Meropenem, isn't that a beta-lactam antibiotic, like penicillin?
Yeah.
But I thought those weren't very effective against TB because of that thick cell wall.
You're right, most beta-lactams aren't that great against TB, but meropenem is a bit different. It's a carbapenem, which is a subclass of beta-lactams, and it has better penetration into the bacterial cell.
Ah, so it can actually get past those fortress walls. But what about clavulanate? What's its role?
Well, M. tuberculosis produces an enzyme called beta-lactamase, which can break down beta-lactam antibiotics.
So the bacteria have their own antibiotic-destroying weapon.
Exactly. But clavulanate is a beta-lactamase inhibitor. It basically distracts the beta-lactamase enzyme, allowing meropenem to slip through and do its job, disrupting cell wall synthesis.
Clever strategy, like sending in a decoy to clear the path for the main attack. What other innovative approaches are in the pipeline?
Another promising area involves nitroimidazoles, drugs like PA824 and OPC67683.
Nitroimidazoles, weren't those used for anaerobic bacteria and parasites?
You're right, but it turns out they also have activity against TB. They work in a really interesting way. They basically act like Trojan horses, entering the bacteria and then being activated to release these damaging molecules called NOx species.
NOx species. Refresh my memory on those.
They're these reactive nitrogen and oxygen species that can really mess up essential cellular processes like DNA replication and respiration. And what's really cool is that these nitroimidazoles work in both aerobic and anaerobic conditions.
So they can attack the bacteria no matter what their oxygen situation is.
Yeah.
That's huge, especially considering TB can survive in those low oxygen environments, like inside granulomas.
Right. It makes them particularly effective against those persister cells that can evade traditional antibiotics.
This is getting exciting. What other novel targets are scientists looking at?
Well, how about targeting the bacteria's proteasome?
The proteasome. Isn't that involved in protein degradation? What does that have to do with killing bacteria?
The proteasome is basically the cellular machine responsible for getting rid of damaged or misfolded proteins. It's crucial for maintaining cellular health. And in bacteria, it plays a really important role in stress response and survival, especially under tough conditions like nutrient deprivation or antibiotic exposure.
So by inhibiting the proteasome, we could potentially make the bacteria more susceptible to stress and other antibiotics.
Exactly. And there are already several proteasome inhibitors being used to treat cancer. Now, researchers are looking at repurposing some of those drugs for TB treatment.
That's pretty clever. So we're essentially hijacking the bacteria's own garbage disposal system and turning it against them. What other cellular processes are being targeted?
Another interesting approach is inhibiting ATP synthase.
Wait, ATP synthase, the enzyme that makes ATP, the cell's energy currency.
You got it. No energy, no life. It's a pretty fundamental principle.
I get that, but how do we target bacterial ATP synthase without messing up our own cells?
That's the tricky part, of course. But there are some key differences between bacterial and human ATP synthase. Researchers are trying to exploit those differences to develop selective inhibitors.
So it's like finding a unique lock that only fits the bacterial key. Very cool. Are there any specific drugs in development that target this pathway?
Yes, there's a drug called bedaquiline, or TMC-207 for short, that has shown some really impressive activity against tuberculosis, even drug-resistant strains. It specifically targets bacterial ATP synthase.
Bedaquiline. I'll have to look into that one. Sounds promising. Are there any other unconventional targets on the radar?
There's actually a fascinating story about going back to the future with a group of molecules called gracelomycins.
Gracelomycins. That name doesn't ring a bell.
They're actually an older class of antibiotics that were discovered a while back but kind of got overshadowed when the rifamycins like rifampin came along. But now they're making a comeback, and for a very cool reason. They target the sliding clamp of DNA polymerase.
The sliding clamp, what's that?
It's a protein that helps DNA polymerase, the enzyme that copies DNA, stay attached to the DNA strand during replication. Basically, it makes sure DNA replication happens smoothly and accurately.
So by targeting the sliding clamp, these gracelomycins are messing up the bacteria's ability to copy its own DNA.
Exactly, and that's a brand new target for TB drugs. Plus, they've shown good activity against drug-resistant strains, which is always a good sign.
That's encouraging. Any other unique targets being explored?
Yep. Researchers are also investigating non-quinolone gyrase inhibitors.
Hold on, didn't we already talk about fluoroquinolones, which target DNA gyrase?
We did. But these new inhibitors work differently. They bind to a different spot on the enzyme. That could be a way to overcome resistance to fluoroquinolones.
I see. So it's like finding a new way to break into the house, even if the old key doesn't work anymore. Clever. Anything else?
Remember those chambered proteases we talked about, like the proteasome?
Yeah.
Well, there's another one called CLPP that's really important for tuberculosis survival.
Okay, I'm with you. How are we targeting this CLPP protease?
There's this amazing molecule called lasomycin, a cyclic peptide that actually activates CLPP. But here's the catch. It causes it to go haywire.
Wait, activate it. Wouldn't we want to inhibit it?
Normally, yes. But in this case, overactivation leads to uncontrolled protein degradation. It's basically a death sentence for the bacteria.
Wow. So we're turning the bacteria's own recycling system against it, forcing it to self-destruct.
It's a perfect example of how we can exploit the bacteria's own biology to fight it. And it really underscores how important it is to understand the fundamentals of tuberculosis in order to develop effective treatments.
Absolutely. It's not just about throwing drugs at the problem. It's about really understanding the enemy, knowing its weaknesses, finding those Achilles heels.
Right. And that brings us back to Chapter 13. It gives us a great overview of TB treatment and all the research that's going on to combat this ancient foe. It's a must-read for anyone in infectious diseases.
Definitely. But beyond the science, there's another layer to this story, wouldn't you say? The human element.
Absolutely. TB isn't just a scientific problem. It's a global crisis with real social and economic consequences. It affects vulnerable populations the hardest, especially in low- and middle-income countries.
So it's a battle against poverty, inequality, and injustice. These factors create the perfect environment for TB to spread.
Exactly. And it just shows that scientific progress isn't enough. We need to address those underlying social determinants of health that make people susceptible to TB in the first place.
It's a complex problem, for sure, But what steps are being taken to address these broader issues?
Well, there's a growing understanding that we need a multi-pronged approach to TB control. Organizations like the World Health Organization are working hard to implement comprehensive programs that focus on early diagnosis, effective treatment, and preventative measures like vaccination.
So these programs aren't just about handing out pills, they're about education, outreach, and community engagement.
Exactly. It's about breaking the cycle of transmission, reducing stigma, and creating a supportive environment where people feel empowered to seek help.
Sounds like a huge undertaking. Are we making any progress?
We are, actually. Global efforts have led to a significant drop in TB incidence and mortality rates in recent years. But the emergence of drug-resistant TB is a major setback. It's a reminder that the fight is far from over.
It's a constant race against a resilient foe that's constantly changing.
That's why we need ongoing research, innovation, and collaboration. We have to stay one step ahead of this bug.
Well, this has been an incredibly eye-opening journey into the world of tuberculosis treatment. We've covered a lot of ground, from the basic biology of tuberculosis to the latest and greatest in drug development.
We've talked about the mechanisms of action of all these different drugs, the challenges of resistance, and the importance of addressing the social and economic aspects of this global epidemic.
It's a complicated story, but it's a story of hope and the pursuit of knowledge.
Right. And for our listeners, remember, knowledge is power. The more you understand about TB, the more you can contribute to the fight against it.
And on that note of hope and empowerment, we want to leave you with this final thought, a little poetic reflection on the people who are fighting this ancient disease.
In shadows they dwell, these warriors of light, fighting a foe, unseen but with might. They toil and they strive, with hearts strong and bold, to break the chains of TB, a story yet untold.
Until next time, stay curious, stay informed, and stay engaged in the fight for global health. You know, it's amazing, right? We've made so many strides in medicine, but TB is still a major global issue. It really highlights that infectious diseases aren't just about the bugs themselves, but the social and economic factors at play.
That's a key point the chapter makes, for sure. Poverty, overcrowding, limited health care, these are all breeding grounds for TB. It really underscores how interconnected health and social justice are.
Absolutely. And it's not just about developing new drugs, but addressing these underlying issues that fuel the spread of TB.
Exactly. We need a multi-pronged approach. The WHO, for instance, is pushing for a comprehensive strategy, strengthening healthcare systems, improving living conditions, addressing those social determinants of health.
So it's about providing access to good nutrition, clean water, safe housing, really empowering communities to break the cycle of disease.
Precisely. It's about creating a world where everyone can thrive, a world without the threat of TB.
And while we work towards that future, there's still so much we can do now. Chapter 13 really emphasizes the importance of early diagnosis and effective treatment.
No doubt about it. Early detection is key. The sooner we can identify and treat someone, the less likely they are to spread the disease. And speaking of treatment, we've talked about all these different drug regimens. But there's one more piece of the puzzle: adherence.
Ah, yes, adherence. It's a classic challenge in medicine. You can have the most effective drugs in the world, but they won't work if people don't take them properly. So what are some of the things that affect TB treatment adherence?
Well, there are a few. The sheer duration of treatment is one. TB regimens can last for months, sometimes even years. That requires a lot of commitment from patients.
I can only imagine. To stick to a strict medication schedule for that long, especially while dealing with life's other challenges, must be incredibly difficult.
Absolutely. And then there are the side effects. Some of the drugs can cause nausea, fatigue, even more serious reactions. It can lead people to stop treatment before they should.
I understand that, but it's so risky. Missing doses or stopping early can lead to drug resistance, making the infection even harder to treat.
Exactly. That's why patient education and support are so important. Healthcare providers need to really emphasize the importance of completing the full course of treatment.
So it's about empowering patients, giving them the knowledge and support they need to succeed. But beyond individual efforts, what about technology? Can it play a role in improving adherence?
Oh, absolutely. The chapter talks about some exciting developments in digital health. Things like mobile apps that send reminders to take medication, track progress, even connect patients with doctors virtually.
That's incredible. Like having a virtual TB coach right in your pocket. Are there any other technological innovations on the horizon?
There are, actually. Researchers are developing new diagnostic tools to detect TB quickly and accurately, even in remote areas. And there's a lot of interest in creating shorter, less toxic treatment regimens, anything to improve adherence and reduce the risk of drug resistance.
It's great to see science and technology being used to fight this ancient disease. But even with all these advancements, there's a sense of urgency, isn't there? Especially with the threat of drug-resistant TB looming.
Absolutely. The rise of MDR and XDR-TB is a wake-up call. We can't afford to be complacent. We need to keep investing in research, innovation, and global collaboration.
Well said. This has been an incredible journey into the world of TB treatment, from the tiniest bacterium to the global fight against this disease.
We've explored the mechanisms of resistance, the social and ethical dimensions. It's been a real deep dive.
And through it all, the power of knowledge stands out. The more we understand about TB, the better equipped we are to fight it.
I couldn't agree more. Knowledge is the foundation of effective action. And as we wrap up this deep dive, I'd like to leave our listeners with a question. What role can you play in the fight against TB?
That's a great question. We can all make a difference, whether it's staying informed, advocating for change, supporting research. Every bit counts.
The fight against TB is a collective effort. And as we've learned from Walsh and Winsvich, it's a battle that's far from over. But with continued dedication and innovation, we can overcome this ancient foe.
On that note of hope and resilience, let's end with this final thought.
A tiny bacterium, a mighty foe. A global challenge, a seed we sow. Of hope and action, knowledge our guide, together we'll conquer side by side.